A neuron is a type of cell that is covered by a membrane, which we know as the phospholipid bilayer. It's essentially a phospholipid bilayer. It's around this whole neuron, but you cannot see it because I did not make one. But within the phospholipid bilayer are protein channels that allows chemicals to enter and leave the cell. While the neuron is at rest, the inside of the cell and the outside of the cell are oppositely charged. As you can tell, the inside is more negative, the outside is more positive. And the negativity of the inside of the cell is due to the negatively charged proteins inside of the cell. The inside of the cell is typically around negative 70 millivolts. This is technically what we call polarization and resting potential. The membrane allows some chemicals in, such as oxygen, carbon dioxide, urea, and water, without the use of ATP, but does not allow other chemicals in without using ATP, such as sodium, potassium, calcium, and chloride, due to its selective permeability. The membrane channels must be stimulated in order to open and allow certain molecules to enter. The sodium-potassium pump is always pumping 3 sodium ions out and 2 potassium ions in through active transport via ATP or energy. Due to the sodium-potassium pump, there are more sodium ions outside of the cell than there are inside of the cell, and there are more potassium ions inside of the cell than there are outside of the cell. Sodium wants to enter the cell because of the concentration gradient. There are more sodium ions outside the cell, so it naturally wants to move into the cell. But sodium also wants to enter the cell because of the electrical gradient. The sodium ion is positively charged, and the inside of the cell is more negatively charged than outside where the sodium ions are. So it naturally wants to move into the more negative environment because it's more positive out here. Let's see. At rest, no sodium ions cross the membrane except through the sodium-potassium pump. And once they are out and they are pumped out, they do not go back in because of the selective permeability of the membrane, the phospholipid bilayer membrane. But some potassium ions at rest slowly leak out of the cell via potassium linkage channels and take a positive charge with them. So they're just seeping out a little bit at a time, all while the sodium-potassium pump is taking 2 in. Potassium is pumped into the cell because it is more negative than sodium is outside the cell, but flows out of the cell because there's more potassium inside of the cell. So it flows out due to the concentration gradient, but goes in via sodium-potassium pump because of the electrical gradient. While potassium ions slowly seep out of the cell while at rest via potassium linkage channels, the sodium-potassium counteracts this and works to bring 2 potassium into the cell. The resting potential stays stable until the neuron is stimulated. Stimulation takes place at the synapse. So there's the synaptic cleft, and here's also a synaptic cleft because the neighboring neuron in bulb would be right here. And there's the dendrites. So, messages sent by axons are called axon potentials. And what happens is neurotransmitters attach to the dendrites, and there's this collection of positive charges at the axon hillock. until the threshold is met at about negative 55 millivolts. A little amount, well, the membrane is depolarized, as we have here, a little bit at a time until the threshold is met. Then a massive depolarization occurs, and essentially an action potential occurs. So, after it, we have this positive charge move into the axon hillock. We have an action potential, essentially, once it reaches the negative 55 millivolts. But the amplitude and velocity of the action potential are independent of the intensity of the stimulus that initiated, provided that the stimulus reaches the threshold. The amplitude, velocity, and shape of action potentials are consistent over time for a given neuron, but thicker axons can convey action potentials at greater velocities, can also convey more action potentials per second. The axon channels that regulate sodium and potassium are called voltage-gated channels. The permeability depends on the voltage difference across the membrane. So, as the membrane is depolarized, so it's being depolarized as it flows down, both potassium and sodium voltage gate channels open and allow freer flow of the ions in and out of the cell. So, they all are open now. So we have our sodium and our potassium pumps, our channels open. There's a rush of sodium into the cell once we reach the negative 55 millivolts. However, opening of the potassium channels does not produce a very significant effect, but the opening of the sodium channels produces a rather large effect because of the tendency of both the electrical and concentration gradient to push the sodium ions into the cell. So there's this big influx of sodium ions as soon as we hit the negative 55, which is threshold. So we have a whole bunch of these sodiums going in. Then we have what is known as a propagation of an action potential. So the positive charge flows down the axons and opens voltage-gated sodium channels among the nodes of Ranvier. So this is a node, this is a node, this is a node, this is a node, until it reaches the end bulb. Once the positive charge reaches the end bulb, neurotransmitters are released. So we have our neurotransmitters. Nodes of Ranvier are parts of the axon not covered by myelin. This is our myelin. Myelin helps the electrical message flow down the axon at a faster rate. Due to the myelin sheath, voltage-gated ion channels are not being opened at every point along the axon. The channels are only being opened at the myelin-free part of the axon known as the nodes of Ranvier. So they're only being opened here, 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 here. And at the peak of the action potential, at about 30 millivolts, positive 30 millivolts, the sodium gates are shut. So we no longer have our blue channels. They're shut. While the potassium gates remain open and the K plus ions are flowing out of the cell at a faster than normal rate. So now the K plus are moving out through the leakage and through the potassium gates. The cell then enters a refractory period, during which it resists the production of further action potentials. During the first part of the refractory period, called the absolute refractory period, the membrane cannot produce another action potential regardless of the stimulation. During the second part of the refractory period, or the relative refractory period, it takes a stronger than usual stimulus to initiate an action potential. So, the inside of the cell is now more positive and potassium ions want to go out. As stated before, the potassium gates remain open. Potassium ions are driven out of the cell due to the electrical and concentration gradient. When the potassium ions leave, they take with them a positive charge. Because enough of them flow out of the cell, a temporary hyperpolarization takes place. Hyperpolarization is essentially an increased polarization beyond resting potential. The inside of the cell becomes even more negative than usual and usually goes to negative 80 millivolts. At the end of this process, the K plus gates close and the sodium-potassium pump works at a not so timely fashion to bring the membrane back to resting potential at negative 70 millivolts. And here we are back where we started.